three subphases. What are they, Nana? G1 phase, S phase and G2 phase. First, when we have to talk about G1 phase, which is the lengthiest phase of interphase. Okay, what happens here? Most of the cell organelle duplication, okay, most of the cell organelle duplication happens in which phase? G1 phase. All the enzymatic machinery required for DNA replication synthesizes in which phase? G1 phase. When we have spoke about those cells which rarely divide, they will escape G1 phase to G0 phase, again they will come back to G1 phase. So that G0 phase is known as what? Quiescent stage or quiescent center. Okay, name few cells, name few such cells, heart cells and nerve cells. And G1 phase time period will be variable. Those cells which frequently divide will have shorter G1 phase. Those cells which divide lately or rarely they divide, they will have lengthiest G1 phase. This is all about G1 phase, you have to remember. And one more pointer, after G1 phase is also addressed as post M phase, pre-synthetic phase. Next phase is what? S phase. Now it will be little easy to remember. So S phase is also known as what? Post G1 phase, pre-synthetic, pre-G2 phase. So in S phase, inside a nucleus what is happening? DNA replication. A hypothetical statement that only DNA replicates but not chromosome. Chromosome number remains same. Okay, on that you got 2 PYQs on that concept only. Next, what happens with a centriole duplication? That happens where? Inside cytoplasm. Like how? If I have a single centrosome after G1 phase, if I have a single centrosome, how many centrosomes will be after S phase? 2 centrosomes. Here 2 centrioles, after S phase how many will be? 4 centrioles. Here 2 chromosomes, after S phase how many chromosomes will be? 2 chromosomes. Chromosome number will be same. Okay. Next is G2 phase. In G2 phase, semi-autonomous cell organelles will duplicate like chloroplast, mitochondria and Golgi bodies. Golgi bodies are not semi-autonomous, but it also duplicates along with this. What protein synthesis will happen at G2 phase? Tubulin protein. What protein synthesis will happen at S phase? Histone protein synthesis. So this is all you have to remember about interphase. Can you remember the synonyms we have discussed for interphase? Interphase is also known as preparatory phase or biosynthetic phase. Preparatory phase or biosynthetic phase. How much time it will consume for complete cell cycle? More than 95%. Like that, for a complete cell cycle, it is variable for every cell. Yeast cell takes 90 minutes. Human cell 24 hours. In that only 1 hour is allotted for M phase. Coming to M phase, first what happens? Karyokinesis, that is nuclear duplication. Okay, synthesis of nucleus might be, or genetic material might be, or movement of nucleus. Any of these definitions is correct only for karyokinesis. In 4 stages it will happen, or 4 phases. What are they? Prophase, metaphase, anaphase and telophase. Okay, so before entering into karyokinesis, we have learnt 3 types of M phases. What are them? 3 types of M phases. Mitosis, amitosis, meiosis. Mitosis, amitosis and meiosis. Amitosis happens in what type of cells? Unicellular cells, be it prokaryotic or eukaryotic. Amitosis is also addressed as what? Binary fission. Okay, so this is addressed as amitosis because there is no involvement of spindle apparatus or spindle fibers. Next is what? Mitosis. In mitosis, first prophase. What you have to remember here? Chromatin material gets untangled to chromatids, which are thread-like structures. They will be adjoined to the nuclear membrane. So it is known as pyreme stage or Bouquet stage. two centrioles which are duplicated in the S phase, they will move to the opposite poles in which phase? Prophase, as asters, and they will start radiating out what? Spindle fibers. This is all you have to remember with prophase. Any terms related to this you have to choose prophase. Next is metaphase. Okay. In metaphase, all the univalent chromosomes will align to a equatorial plane. This phenomena is known as what? Congression. Which part of the chromosome is attached to spindle fiber? What is the site of attachment? Kinetochores. How many kinetochores are present for a single chromosome? 2 kinetochores. Okay. At the primary constriction, 2 disc shaped structures are there that will be aligned with spindle fibers. And anaphase is the shortest phase. What is the longest phase? Prophase. And in metaphase you can see clear morphology and structure of chromosomes. Right? Sit straight all. That enhances your memory. Anaphase. Anaphase is what? Longest phase or shortest phase? Shortest phase. Here what happens? Centromere splitting happens. Centromere splitting, and chromosomes will move to the opposite poles with different shapes. Can you tell me those shapes? I shape is telocentric and J shape is if I say you will change the word. Acrocentric. L shape submetacentric. V shape metacentric. Okay. And where do you see P arm and Q arm? P arm and Q arm. In which type of chromosomes you see? Which shapes? Acrocentric and submetacentric. You will see P arm and Q arm. Amongst these two arms of chromatid, tell me which arm is longest and which arm is shortest. Q is long, P is short. We will write Q like this, no? Like that you remember. Long face. Okay. Long arm. So, this is the shortest phase where you can see clear shapes of the chromosomes. And tell me, in these two phases, where you can see elongated spindle fibers? Metaphase. Shortest spindle fibers? Anaphase. Last is telophase. Here at the end of prophase, we will address it as late prophase. Nuclear membrane, nucleolus, endoplasmic reticulum, Golgi bodies will disappear. Means they are not visible under the microscope. As such, cytoplasm will get condensed. Here they will reappear. They will gain back their structural stability. Okay. They will reappear here, and here chromatin gets untangled to chromatids. Here chromatids are getting tangled back to chromatids. Clear, right? This is about karyokinesis. And then when it comes to cytokinesis, we have discussed it very clearly, no, how it happens in a plant cell and animal cell. First we will discuss animal cell. It is very simple, right? A furrow will be formed from the periphery, and then karyokinesis, cytokinesis will be operated via spindle fibers. Karyokinesis is operated, cytokinesis is formed via cell furrow. But there is a process involved in cytokinesis for plant cell. Why? Because assertion reason it is important. Why cell plate is forming at the center? Why cell furrow is not coming from the periphery? It is because it has tough cell wall outside. It cannot form the cell furrow. So, cytokinesis is operated from center. Here it is from periphery to center. So, this is known as centripetal mode of cell wall or centripetal mode of cytokinesis. This in a plant cell, if you see, at the center at first what is formed? Phragmoplast, which is synthesized by phragmoplast. Very good. It is rich in calcium pectate and magnesium pectate. But which organelle is synthesizing phragmoplast? Golgi bodies. And then this phragmoplast will move to the peripheral ends, forming what? Cell plate. Cell plate forms the primary cell wall. That is what? Middle lamella. This middle lamella will give to neighboring cells. Okay, this will fix to neighboring cells. This is a common cell wall between two neighboring cells. Okay, this is about middle lamella. Inner to middle lamella, a bit of cell unit of life will remember. Inner to the middle lamella, what is formed by? Primary cell wall. Inner to primary cell wall, secondary cell wall. Secondary cell wall is close to cell membrane. Right. So this is how cell plate is formed. As it is from center to periphery, this is known as centrifugal mode of cell wall formation. Centripetal, centrifugal, you have to remember. Then getting back to meiosis, which is also known as what? Reductional division. And significance of mitosis we have learned, right? Mitosis takes place in diploid cells of animal cells. The only exception you can take is what? Honeybees, where haploid cells undergo mitosis. In plant cells you can see both haploid cells and diploid cells are undergoing mitosis. And then about meiosis, they help in metamorphosis. Significance of mitosis: metamorphosis, regeneration of lost tissues, okay, and it will maintain the nucleocytoplasmic ratio of the cell, right? These statements please remember. Then meiosis, if we see, we have meiosis 1 and meiosis 2, right? Meiosis 1 we again have what? Karyokinesis 1 and cytokinesis 1. Karyokinesis 1 has how many substages? 4 substages. What are they? Prophase, meta, anaphase and telophase. Prophase 1 has 5 substages in it, which are leptotene, zygotene, pachytene, diplotene, diakinesis, right? And then in between there is a stage, what is it called as? Interkinesis. Is there any organelle duplication happening here? What is that? Centriole duplication happens. Does DNA replicate? No. No DNA replication. Cell is metabolically active. Centriole duplication happens in an animal cell, right? And then DNA and protein synthesis will take place at interkinesis stage. DNA and proteins whichever are required, RNA and proteins whichever are required for meiosis 2 will be synthesized in interkinesis. Meiosis 2 is same as that of what? Mitosis. But there is only one difference. This operates in germ cells, that operates in somatic cells, right? So here, if I have to see, mostly the questions are asked from what? Prophase 1, and it is important actually, hotspot of cell division. So here in leptotene, what are the changes that you can see? Chromatin gets untangled to chromatids, centrioles move to opposite poles, spireme and bouquet stage you can see, that's it, and the chromatids are visible under what microscope? Light microscope. Zygotene is the stage where homologous chromosomes are formed. Synapsis is seen between what? Homologous chromosomes. What are homologous chromosomes? In your words, we have discussed yesterday, no? Huh? Okay, came to nearest point. In your own terminology, we have discussed, right, what are homologous chromosomes. Any two chromosomes which has the alternating forms of alleles of a single gene are aligned close to each other. For example, homozygous dominant black hair, let us consider, heterozygous dominant black hair, homozygous recessive brown hair. Hair color is a trait we have considered, and these are different alleles, and these are the set of chromosomes that are present. There are many chromatid threads in the leptotene stage. In zygotene stage, all the different chromatid threads which have a single traits on two alleles, like genes on the same locus. For example, here capital B, small b, small b. The alleles of a single gene are present at the same locus on different chromatids. These are known as what? Homologous chromosomes. So what are homologous chromosomes, ma? The alleles, alternating forms of a gene are what? Alleles. Alleles of a single gene present at the same locus on different chromatids or chromosomes are known as what? Homologous chromosomes. We have two sister chromatids and two non-sister chromatids of homologous chromosomes, right? So this phenomena is known as synapsis, and this complex is known as what? synaptonemal complex. As such, what are formed? Bivalents are formed in the zygote. What are they? Bivalents. Single bivalent has how many chromatids? 4. 4 chromatids, right? So, one bivalent is equal to one tetrad. One bivalent is equal to one tetrad. Clear? This is all you have to remember on zygote. Bivalent formation, tetrad occurrence, synaptonemal complex, homologous chromosomes formation. Okay, any of these terms belong to what substage? Zygote, right? And then about pachytene. Pachytene is the phase where recombination occurs. Now you have same alleles at the same locus, easy for crossing over, which is what mediated process? Enzyme mediated process. What is the enzyme which is mediating? Recombinase. That is forming what? Recombination nodules, right? And then diplotin stage. So now recombination happened. As such, what shaped chromosomes are formed at the recombination spots? X-shaped chromatids are formed. And this is what? Dissolution of synaptonemal complex, yes or no? Synaptonemal complex is like this. After crossing over, it is like this. So there is the diplotin is the stage which is beginning with dissolution of synaptonemal complex, right? And then you can see X-shaped structures or formation of chiasmata. And this is the stage where mammalian oocytes will stop their cell division for years long. Okay, that is why this is also known as what, ma? Dictyotene stage, right? This is about diplotin. Last substage of prophase 1 is what? Diakinesis. Here this phase begins with what, ma? Terminalization of chiasmata. X-shaped chromosomes will be terminalized, and this begins with terminalization of chiasmata. And here at the end, nuclear membrane, nucleolus, Golgi will be disappeared. Then metaphase 1. What is the difference with mitosis? Here it forms two sets of chromatids on two equatorial plates. So this is forming a bimetaphasic plate. The sister chromatids of homologous chromosomes are aligned here, right? And then anaphase 1. What is the difference, Ra? There is no centromere splitting. Anaphase complex protein is inactive. What is anaphase complex protein? That will initiate the centromere splitting, which is operated in mitosis, but that is inactive in meiosis 1. Okay, wherein the sister chromatids of homologous chromosomes are moving to opposite poles, right? There is no centromere splitting. And then telophase 1. Telophase 1, if you see, the chromatids are formed and two daughter cells are seen here. Again the same story like how it happens in the mitosis. And here you have to tell me, are these haploid daughter cells or diploid daughter cells? After meiosis 1, you will see haploid or diploid daughter cells. Haploid daughter cells we have learnt yesterday, right? Okay, see, I will tell you how. If you suppose this as a cell, right? So here you can see two metaphasic plates, right? Let us suppose asterisk present here and here, aligned like this, right? Bimetaphasic plate where? In metaphase 1. In anaphase 1, can you see here, these two chromosomes are moving to this pole, sister chromatids are moving to the either poles, non-sister chromatids are not moving. So these two sister chromatids will move to the upper pole and these sister chromatids to lower pole. Okay. So can you see any centromere splitting here? No. But as chromatids are moving to the extreme poles, here itself we will count it as two different cells, cell 1 and cell 2. Here in a single cell, how many chromosomes are there? 4. 4, right? Here in a single cell, how many are there? 2. So there is reduction in chromosome number. Here you can see complete set of chromosomes, here you can see half the set of chromosomes. So chromosome reduction is happening in which phase? Anaphase 1, which is producing what cells after telophase 1? Haploid daughter cells. Fix it in your mind. Okay, clear, right? And then interkinesis stage. No DNA replication, cells are metabolically active. In animal cells, centriole duplication takes place, right? RNA and protein synthesis occurs. Meiosis 2, same as that of mitosis. And here there are single chromosomes, right? So you can see univalent chromosomes. Whereas in metaphase 1 you can see bivalent chromosomes. Here univalent chromosomes on the same plate. Anaphase 2, centromere splitting occurs. ACP protein is activated, right? And chromatids are moving to the opposite poles. Telophase 2, you can see the same features which happen in every telophase, where the organelles will be gaining back their structural stability and chromatin is formed back. As such, at the end, how many daughter cells are formed? 4 daughter cells which are haploid, right? So this is all about meiosis. Few calculation part that we have to remember, because we will get questions on the ploidy, okay, like connecting with the chapters like sexual reproduction of flowering plants might be, plant kingdom might be, the ploidy questions will be asked, because we have alternation of generations, right, for all the 5 divisions of plant kingdom: algae, bryo, pterido, gymno, angio. You should know which structures are falling under gametophytic phase, which structures are forming under sporophytic phase. If you have idea on ploidy, you can solve the questions from mitosis and meiosis especially. So to calculate number of cell divisions that occur in mitosis. For example, 100 cells are formed, 100 daughter cells. What is the formula? Formula to calculate is n minus 1, okay, where n is number of daughter cells formed. So 100 minus 1 is how much, ma? 99. 99 mitotic divisions are required to form 100 daughter cells, okay. And then how to calculate meiotic divisions? Here if you observe, number of daughter cells formed, for example 100 cells are formed as a result of meiotic divisions. How many meiotic divisions are required to form 100 cells? It will be n by 4, which is 100 by 4. 25 meiotic divisions are required, right? So this is how you will calculate mitotic divisions and meiotic divisions. And then how many divisions are required for the formation of seeds, fruits and grains? When seeds are formed as a result of fertilization, right? So if you observe in sexual reproduction of flowering plants, we have learnt, first from female we will consider. In a female or in a gynoecium, inside ovary we have ovule, inside ovule we have nucellus. One of the nucellus cell at micropylar region will turn to megaspore mother cell. That megaspore mother cell will undergo meiosis, forming how many megaspores? 4 megaspores. Able to recall? 4 megaspores. Those 4 megaspores are aligned in the linear fashion, of which 3 will degenerate, 1 megaspore is functional, right? So though meiotic division is providing 4 megaspores, of that how many are functional? 1 megaspore. So for under females we have to consider only 1x, 1 megaspore is only functional. But then in males, that is pollen grain is the male gametophyte, right? How is it formed? From microspore mother cell. Okay, one microspore mother cell, after undergoing a meiotic division, it will form 4 microspores. 4 microspores will develop into 4 pollen grains. So here we can apply the same formula, that is x by 4 or n by 4. This two will unite forming female gametophyte, male gametophyte will be releasing what? Female gamete and male gamete. As a result, zygote is formed that matures to what? A seed. So for example, 100 seeds are formed. How many meiotic divisions occur to form 100 seeds? The formula is what? X plus x by 4. If you just remember this formula, it is enough. But you have to know, right, why are we taking this formula? Okay. So it is what? 100 plus 100 by 4. What will be the answer? That's all. Okay, clear now till here? This is how you have to calculate on mitotic, meiotic and this seeds, fruits or grain formation. How many meiotic divisions are required? If you are clear on this, we will move on to NEET PYQs. Done? Yeah. Let's get started. Drink some water and sit. Team A and Team B. We'll give first chance to them this side. Huh? I need a paper and pen. Yeah, pen is there, paper also is here, no problem. Huh? This page is enough. Yeah, to this first question, which is already asked or what? Yeah. Though it is an easy question, take 1 minute time and answer. Okay. Just recall everything, read all the sentences, read the question completely, interpret properly and answer. You can say, if you are ready with the answer, you can say. Yashwant, you are ready? Then say. Option 1. So here in G1 phase, yeah, G1 phase, cell is metabolically active and continuously grows but does not replicate the DNA. Okay, correct. And then S phase, synthesis of DNA occurs and amount of DNA per cell will double. Next, G2 phase, proteins are synthesized and cell growth continues. M phase, actual cell division occurs. Done. Next, 1 second. Let us recall few more points in this. What protein synthesis takes place in G2 phase? Tubulin. In S phase, along with DNA replication, what duplication happens? What organelle duplication happens in cytoplasm? Centriole duplication. If at all there are 26 chromosomes at the end of G1 phase, how many chromosomes will be at the end of S phase? Same, 26 chromosomes. And then G2 phase, what are the organelles that are getting duplicated? What organelles are getting duplicated in G2 phase? Mitochondria, chloroplast, Golgi. Which is the lengthiest phase amongst this? Second question. What is the right answer? Spindle fibers attach to kinetochores of chromosomes during. Think and answer. Be confident on your answer. What is the answer? Metaphase. Spindle fibers attach to kinetochores of chromosomes during metaphase. One small question again. During mitosis, you will find univalent chromosomes. So, two kinetochores of a chromosome are connected to the spindle fibers. Whereas in meiosis 1, how many kinetochores of a chromosome are connected to the spindle fibers? For a single chromosome, there are two kinetochores. How many of them are connected to spindle fibers in meiosis 1? In metaphase 1? This is mitosis, ma. Univalent chromosomes. Two kinetochores are there. They are attached to the spindle fibers. Right? Meiosis 1. You will see bivalent chromosomes. Right? They also have two kinetochores. But then only one kinetochore of each chromosome is connected to what? Spindle fibers. Just remember the diagrams. That is the reason I will draw a box and do some cartoon diagram here, so that you will remember, right? So, question is twisted in any ways, you should be in a position to answer. Next, this question, this side. Okay. You are saying option 1. Okay. Statement 1 is what? Chromosomes become gradually visible under light microscope during liptoting stage. True. The beginning of diplotin is recognized by dissolution of synaptonemal complex. Good. So, both statement 1 and statement 2 are correct. Can you name few more events in the diplotin? Any few more events in the diplotin? Say it louder, no problem. Louder. Formation of chiasmata, good. What cells will be stopped in the cell division in this stage? Oocytes, mammalian oocytes, right? That is the reason there is also one name given to diplotin. What is this? Dictyotine stage. Okay. So, clear till here? Next question. So, mostly we got questions from cell the unit of life only, ma. Hardly 2 or 3 we will get from cell cycle and cell division, 2 or 3. That also sometimes will be connected with ploidy and they will ask. This side or this side? Who is huh? After anaphase. We have done this question already, right? Yeah. So doubling of number of chromosomes can be achieved by disrupting the mitotic cell division soon after anaphase. How many of you can answer this correctly, or if you are thinking still? How many of you are clear with the answer? You just raise your hands. If not, I will explain this. Everybody are clear? I asked how many of you are not clear, raise your hands. Okay, down. See, anaphase, listen, disrupting of chromosomes. It will not affect the chromosome duplication they are asking, right? In anaphase what happens? Chromatids will move apart, right? Chromatids will move apart. So if you disrupt the cell division after anaphase, do you see any effect on the doubling of chromosomes or centromere splitting? No. The question is as simple as it is. Now you read the question. Doubling of number of chromosomes can be achieved by disrupting mitotic cell division soon after. Okay, so that's all. Next, fourth question. See, this is second time we got the question on 2026 and 2023. Don't expect that easy paper, huh? During which stages of mitosis and meiosis respectively the centromere of each chromosome will split? Second option. Centromere split at what? Anaphase of mitosis and anaphase 2 of meiosis. In anaphase 1, centromere splitting doesn't take place because which protein is inactive? Anaphase complex protein is inactive. So the right answer is option 2. Next question, ma. Second, leptotene. Okay, so what is the right answer? Next question, 2023 again. Which one of the following is quiescent stage of cell cycle? What is that? G0 phase. Cells are metabolically inactive in G0 phase. True or false statement? False. Cells are metabolically active. We have lengthiest G0 phase. True or false? It is variable per cell, you have to say. Okay, it is variable. And from G0 phase, the cells are resuming back their cell division to S phase. False. They will get back to G1 phase again. Next, this question. This one. Which of the following stages in meiosis involves division of centromere? It is anaphase 2 of meiosis. 3 to 4 times the question is asked on anaphase 2 only, anaphase and anaphase 2. Raise your hand and answer. First one, in packitine, right? That recombination nodules are formed between homologous chromosomes or non-homologous chromosomes? Homologous chromosomes of what chromatids? Non-sister or sister? And chopakra. Sister or non-sister? Non-sister chromatids of homologous chromosomes are involved in what? Crossing over. Okay. Enzyme involved here is louder recombinase. Options are given above actually. Just C and answer. First option, M phase, equational division. If they have given, if they are particular or mitosis or meiosis, we can differentiate. But here we can choose equational division. Then what ma? G2 phase. Proteins are synthesized. Next what? Quiescent phase, inactive stage. Next G1 phase, interval between mitosis and initiation of DNA replication. Next. Statement based question, you cannot calculate on air and see. Just have a pen, paper, think about it and answer. First, we'll see. So here the question is about choosing what? Correct answer, means correct options we have to choose. Tetrad formation is seen in which phase? Tetrad formation. Louder. Zygote. In zygote, tetrad, bivalent, homologous chromosomes, synapsis, synaptonemal complex formation. Any of this term you have to choose what? Zygote. It occurs in zygote. Second, during anaphase, the centromeres split and chromatids separate. Terminalization takes place during diakinesis. Very good. Next, nucleolus, GCER reformed during telophase. Crossing over takes place between sister chromatids of homologous, non-sister chromatids. If I keep sister and non-sister chromatids here, what you will choose? Incorrect statement, right? So it is between non-sister chromatids of homologous chromosomes. So B and D. Next. We have many questions to do. Sit straight, be active, drink some water. Third one. Okay, we'll see. Statement 1. During the G0 phase of cell cycle, the cell is metabolically inactive. Cell is metabolically active. Statement 2. Centrosome undergoes duplication during S phase of interphase. Right now? So, statement 1 is incorrect and statement 2 is correct. This. We'll give some time. Which stage of meiosis can last for months or years of oocytes of some vertebrates? Diplotin, right? So that is why it is also known as dictyotin stage. Good. We have covered everything now. Super brains. Without paper, without pen, just like this we'll say. Option 2. 2? Yeah. So we need to choose the correct answer for the options. Identify the correct sequence of prophase 1. First is B, you say Chromosomes become gradually visible under the microscope at which stage? Leptotin. Next, A you are saying synapses of homologous chromosomes in zygote. C, crossing over between non-sister chromatids of homologous chromosomes happens in paquetine. Terminalization of chiasmata, diakinesis. Dissolution of synaptonemal complex, diplotin. So it is B, A, C, E and D, right? Next. Remembered everything, ma? Recalled? Second, synaptonemal complex in zygote stage. You can see bivalent and tetrad. One bivalent is equal to how many tetrads? One tetrad. One bivalent will have 4 chromatids. One tetrad will have 4 chromatids. One bivalent is equal to 1 tetrad. Next. Think and answer. Ma? Let others also think and confirm the option, then we will go with the explanation. With respect to metaphase, which of the following statements is incorrect? Chromosomes lie at equator of the cell. Complete disintegration of nuclear envelope takes place. Chromosomes are highly condensed. Metaphase chromosomes are made up of 4 sister chromatids held together at centromere. It is how many sister chromatids? 2 sister chromatids for a homologous chromosomes, right? And complete disintegration of nuclear envelope takes place where? At the end of prophase. So the incorrect statement is 2 only. But here metaphasic chromosomes are made up of 2 sister chromatids each, right? If it is, if the question is on metaphasic homologous chromosomes, then we can accept this. So directly this is wrong, indirectly this is also wrong. I feel so. Okay. Next. This question. We have many questions to do. Show some energy. The appearance of the appearance of recombination nodules on homologous chromosomes during meiosis characterizes. What is the right answer? Louder, be loud with your answer. First one, right? Where do you see the recombination nodules? At the site of crossing over. Okay. So, if I have to see this as chromosomes, these are the sites of crossing overs. This forms out what? Recombination nodules. Next, this question. Which of the following never occurs during mitotic cell division? Simple, easy to answer. Louder. What is the right answer? First one. Pairing of homologous chromosomes happens in which phase? Pairing of homologous chromosomes happens at which stage? Zygote of prophase 1. Do you see that zygote and prophase 1 in mitosis? No. So what is the right answer? Pairing of homologous chromosomes will never occur in mitotic cell division. Next question. Fourth option. Select the incorrect statement with reference to mitosis. Spindle fibers attach to centromere of the chromosomes. No, it is attaching to kinetochores of the chromosomes. Next, this question, ma. Regarding meiosis, which of the statements is incorrect? Fourth option. DNA replication occurs in S phase of actually interphase. Which of the following stages of meiosis involves the division of centromere? Anaphase 2. It will not happen in anaphase 1. Next, this is fourth time na question on anaphase from 2021 to 26. Fourth time you are seeing. This is some repeated question but asked in 2021. One asked in 2024. So this questionnaire session is to give you an idea on the patterns of questions, on the concepts of the chapter, right? Which is stage of meiotic prophase? Some 10th time on meiosis 1, prophase 1. What is the answer? Terminalization of chiasmata, diakinesis. Next. This is the last question. Okay. This is the last question. Few more are there. We will do it tomorrow. The fruit fly has how many chromosomes? 8 chromosomes in each cell. During interphase of mitosis, the number of chromosomes at G1 phase is 8. What would be the number of chromosomes after S phase? 8. Accepted? Is it 16 or 8? 8. If I have to say 8C DNA at the end of G1 phase, how much amount of DNA after content of DNA after S phase? S phase it will be 16C, but chromosome number will be same, right? Few more questions, we will do it tomorrow, okay? So that's all with cell cycle and cell division. We will start on biomolecules in the next class.